Uh, Hi everyone. Thanks for coming today. It's a wonderful day here in Los Angeles, and it's also a good day in Baltimore, where Jordan Green is resides. So Jordan and I go way back from the time we were early graduate students at MIT, and I'm very happy to have Jordan give a seminar today. Jordan has done incredibly well. He's currently professor of biomedical engineering and many other affiliations, including ophthalmology, neurosurgery, oncology, material science, chemical engineering at Johns Hopkins. And he's also the director of the bioengineering program, the biomedical engineering undergraduate program, which is, as you may have heard, is ranked #1 in the country. He's also very much involved in the Translational Tissue Engineering Center and is the associate director, as well as the Translational Immunoengineering Center at Johns Hopkins. He received his undergraduate and his undergraduate from Carnegie Mellon and his PhD from MIT, and subsequently also stayed at MIT a little bit longer. Until he started, of course, at Johns Hopkins. He's really one of the top young people in the field of biomedical engineering. He is also very entrepreneurial, and he's set up multiple startup companies that have done incredibly well. And he is also very well in academics. He's a fellow of a number of different societies, including the Biomedical Engineering Society, AIMBE, and he's also associate editor for Science Advances, which is one of the top journals in the field. So he also has been extremely prolific and inventive, many publications and an incredible number of patents, and for which all of these he's also received many distinguished recognitions, including the Popular Sciences Brilliant 10, which is a very high recognition, the Presidential Early Career Award for Scientists and Engineers, as well as numerous other very distinguished recognitions. So with that said, I will not continue because I think we've already run way over time, and I do want to give people some time for a question and answer period, but it's a huge honor for me to introduce Jordan, a wonderful friend and an amazing researcher.
Well, thank you so very much, Ellie, for the invitation, a very kind introduction. And it's with all of you. Am I on mute or
No, yeah, I wanted to put myself on mute. I put you on mute. Sorry about that.
Okay, but it's a great pleasure to be here, and thank you so much for the invitation, that kind introduction, and to share with all of you about work we're doing on polymeric nanoparticles for cellular programming and how this can enable precision and personalized medicine. And then just also a shout out to Hopkins BME, to those that are out there looking for programs to apply to for undergraduate or graduate studies. You know, definitely come check us out. And so I want to give some snippets of some different stories that are all related to the same theme, which is how we can have new types of enabling technology, advanced therapy that involves intracellular delivery, and how that can open up a lot of opportunities with genetic medicine and precision medicine and personalized medicine. And so first, to focus in the eye, and so I have an appointment in ophthalmology in the Wilmer Eye Institute, and I'm very interested in blindness and ways that we can combat that. And so here's, I wanted to share with you some of our recent work on this. And so from a big picture, what we're looking at is gene therapy, is intracellular delivery, and then there's different approaches to this problem. And a lot of the approaches are based on viruses and engineering viruses. And what we're interested in instead as biomedical engineers is to look at how we can use non-viral approaches that can be much safer, so we don't have any intrinsic problems with toxicity or immune response or carcinogenicity, but then also enable a lot of things that a virus can't do. For example, to have very large cargo capacity where we can mix and match genes just by simple mixing, and there's no limit to the size of what we deliver. It's very easy to scale up, and it's easier then to provide accessibility to these therapies for patients. The challenge is, how do you do it safely and effectively? And a lot of materials have fallen short. A lot of conventional non-viral materials like conventional polymers have fallen short. And so how do we look at this in a new way to have improved intracellular delivery? And so we have to look at each of the steps in the delivery pathway. And so the first step is that we design a material, in this case, it's a positively charged polymer, to bind the negatively charged nucleic acid cargo, the DNA or the RNA, compacted into a particle that protects that cargo from degradation and facilitates uptake in the cells. And so this could be ligand specific, or it could be biomaterial mediated, that it goes into specific cells. Once inside the cell, a lot of delivery materials, a lot of types of nanoparticles actually get trapped in endosomes. A lot of lipid-based and liposome materials get stuck in endosomes. And in the endosome, it's kind of like when we eat food and we digest it in our stomach. When a cell eats the particles up into an endosome, it digests it. There's different enzymes there, there's low pH, and it'll degrade the contents. And then often it'll spit it out again with exocytosis. And so we need to design the materials to be able to buffer the endosomal compartment, lead to transient rupture of the endosome, and get out into the cytosol. And then once here, a lot of materials can hang around and cause toxicity or don't efficiently release the material. So we wanted to design the materials that we made, the polymers, to have quick degradation in water, and in some cases even trigger degradation as soon as they sense they're out of the endosome and in the cytosol, a reducing environment. They've triggered break up and release the cargo. And in the case of the DNA, it then needs to make its way into the nucleus to be expressed. And so the approach we've taken is to look at different types of polymers, and we've made different libraries of polymers now for different kinds of function, where they're delivering different cargoes or to different cell types. And a lot of these are based on different types of ester amines, including polybeta amino esters. And what we found is that by changing the small molecule end group of the polymer, it can make dramatic differences to the overall efficacy. And so we have different structural components, like primary and secondary amines to bind the nucleic acid and protect it, tertiary amines to facilitate that endosomal escape that was key, and then degradable linkages to enable release in water over time with the esters. And what we've seen is that we can now have delivery to a lot of different cell types. But then how do we extend that in vivo, and what's the durability look like? And so we've looked at a lot of different structures to explore the chemical space and to understand which types of structures are most effective for delivery. And then what we found is, using this approach, we can get viral-like efficacy in delivering these genes. In this case, we're delivering just reporter genes that then are express fluorescent proteins, to see how they work in different cell types. And a lot of cells are usually seen as very hard to transfect, like you must need viruses to do that. We can actually do it with these biodegradable polymers. And it's just mixing the biodegradable polymer with the DNA, waiting a few minutes and adding it to cells, or we make it lyophilized and then it's just resuspended in water and add it to cells, or add it in vivo. And so it's very simple to use. And the lyophilization, we found this was very important, because since we're designing the materials to be degradable and really quickly degradable, where they can just degrade in hours to a day, then the question is, well, how do we give that to collaborators and clinicians that could be all across the world that they would like to use this as a medicine? And so what we developed are ways that we can have the particles stored in a dry state, and then they're stable long term. And so the trick was to just add sugar. And so by adding sugar as a cryoprotectant and then lyophilizing or freeze drying, we get this powder that then, even if when it gets wet, it degrades really quickly. As long as it's kept dry as that powder, it can be stable for a very long time. And so with our studies so far, we've seen that efficacy stays the same for at least 2 years if they're in this lyophilized state and they have the same nanoparticle properties. And so even though when you add water, it does its thing, very efficient delivery, quickly degrading, very safe, it can stay with a very long shelf life if it's kept dry. And especially if it's kept dry and then also cold. And so what we wanted to do then was to look at the eye. There's the leading causes of blindness in Americans is American adults. It's age-related macular degeneration and diabetic macular edema. And these affect millions, and they affect millions around the world. And it's caused by, in large part, to this overexpression of VEGF. There's all these other diseases that are rarer, but then affect children and affect others all over the world that are due, in many cases, to single gene mutations that then lead to retinal diseases. And so what we wanted to see, is there a broad way that we could address these problems by safely and effectively delivering genes to the eye that then get expressed, and to do it without viruses. And so if we don't use viruses, we could do repeated administrations, we could use large genes, we can use combinations of genes that could really open the field up. And so we're interested in making these nanoparticles. This is what they look like. They're about 100 nanometers in size, the polymer DNA particles. And then while often drugs might be injected intravitreally or subretinally, what we're interested in, and this is together with Peter Compasiero at the Wilmer Eye Institute, was could we look at this new route of suprachoroidal delivery, which is this potential space between the sclera and the choroid. So this is the white part of your eye is the sclera. So it's doing the injection just past that white part. So not going deep into the center of the eye, just on the outside. And it's something where you could do really on the side of the eye. And then the idea is that this volume will get convected and whisked away through this potential space, this suprachoroidal space, all the way to the back of the eye and around the other side. And then you'd have a high concentration right there, instead of injecting it in the middle, intravitreously into this gel, where the particles really wouldn't diffuse that well, or you wouldn't get good delivery. So this was the idea. And so the new things we're doing is we're using non-viral particles, we're using suprachoroidal delivery, and could this approach work? And what we see is that we see widespread expression of GFP in the retina and in the RPE. This is 2 months after injection. And so we inject in one site, but we see transfection all the way around. And so like we had hoped and hypothesized, we were able to get those particles to distribute all the way behind the eye and around the other side to transfect a lot of retina. And then when we look at it over time, even though we know the particles degrade quickly, within hours to a day, what we see is that the plasmid they deliver, which doesn't integrate, it's an episomal plasmid, is then able to have very long life. Because these cells aren't dividing, the plasmid just sits there and it can keep making the protein long term. And so we see after 2 weeks, 1 month, 2 months, 4 months, 6 months, 8 months, we have similar efficacy, really high efficacy that persists over time. And so what this means is you could have one injection that could then last a year or many years, which would really change the standard of care for these diseases, either because you need injections every month right now, or because there's really no factor that works at all. And then if we look spatially, what we see is that just like we had hoped, we do an injection and the particles transport and convect all the way around to the back of the eye and the other side. So we see this good expression throughout the retina, which is really what we want to do with a conventional, well, still very novel subretinal injection of a virus, for example, for gene therapy in the eye, which has been approved. And what you have is transfection in just that area of that subretinal injection, like a bleb. It's not all through the retina in this way. And this is also much safer. This is a minimally invasive outpatient procedure to do this injection. And what we also found is if we do multiple injections, unlike with a virus where you can get an immune response and so it stops working over time, what we want with the non-viral approach is if after a few years you did want another injection, or even in the first few weeks you wanted to boost levels, could you do these repeated injections? And so what we found is if we do more than one injection, like for example, if we inject 3 times, we significantly boost the level of protein. And so we can do these multiple injections and they can have dramatic efficacy. And this is just to see what this looks like again with reporter genes. If we see the bird's eye view that we spread out the retina, this is what we see with the GFP expression, and we zoom in. Even from the zoomed out view, it might not look like everything is transfected, but you zoom in there and you see that just about everything is transfected. We're getting very, very large areas of transfection through the RP, the retina, the choroid. And then if we look at a cross section, we see the same thing. So all the yellow and red is showing GFP expression. And so we see this really strong expression. And so instead of a reporter gene, we could really do anything, but this shows proof of principle of how durable and widespread the expression is with just a single non-viral injection. And that we can do the repeated injections to boost further. And so we then were interested, well, let's try something functional. And so what we did is we delivered VEGF. And so many of you may know how important VEGF is for applications for regenerative medicine and tissue engineering, with providing vasculature to scaffolds and growing tissues. Well, in the eye, it has the opposite effect. because you don't want all these blood vessels to grow in the eye. And then when that happens, that neovasculature obstructs vision, the leakage from the vessels obstructs vision, and this is what causes these leading causes of blindness. But what's needed are new animal models. So in order to treat these VEGF-dependent diseases like AMD and DME, what we really would like is to have improved animal models that could be ways to make the disease look just like a person that has a late stage wet AMD, let's say, but be able to be applicable in rodents or larger animals to induce that very easily. And so the idea was, well, maybe we could use suprachoroidal injection of these nanoparticles to create these models that reproduce wet AMD really well. And so that's what we found. So instead of doing GFP, we're delivering VEGF, but then the green signal here is showing the vasculature and the leakage. And what we see is that these blood vessels are growing and leaking. And this is what you see with a patient with wet AMD, and this is what blocks their central vision and what makes them blind. And so by just doing a single injection of the nanoparticles, we can create these models. And then if we look at them over time, they actually progress to have this subretinal fibrosis, which usually you don't see until the advanced stage in wet AMD patients. And it's very hard for an animal model to show this so that you can try to act on it. But in this system, again, just the simple injection of the nanoparticles, the gene they deliver is very functional. It expresses throughout the retina with good durability. And so we can create these animal models easily. And then what about doing something on the therapeutic side? So we instead used a model that was a gold standard model to look at neovascularization, which is what is the culprit with wet AMD. And in this case, instead of delivering VEGF that causes the disease, we deliver an anti-VEGF agent to treat the disease. And what we see is that with a single injection of that, we do reduce the neovascularization, and that we can do this over time. And so that this can be used as a robust therapy for VEGF-dependent diseases, but more generally, this could be a new platform for treating these rare genetic diseases and other retinal diseases to turn on the factors that would help with healing in a very safe and durable way. So that's delivering DNA and turning things on. What about delivering smaller nucleic acids like siRNA or microRNA and turning things off? And so here I wanted to share with you a cancer application. And so for siRNA delivery and microRNA delivery, the cytosol delivery is critical because in this case, we don't need it to go into the nucleus. We just want to get a big amount in the cytosol. And so what we found is in this step, the releasing the cargo from the particle into the cytosol, could we make this more effective than the previous polymers we were using that were focused on DNA? And so what we found is that if we added disulfide linkages, and it could be throughout the polymer, or it could be just the end group of the polymer, because the end group of the polymer is where the primary amine is. And so if that primary amine falls off, the real charge that's holding the nucleic acid with the polymer falls off and it'll separate. Because again, these are formed by, it's like a magnet. There's a positively charged polymer and negatively charged nucleic acid, and they electrostatically attract each other, the positive and the negative. And so that's what we'd have there with the release. And so if we have an end group that doesn't have a disulfide, then what we see is that this is the ratio of polymer to nucleic acid, the weight ratio. And then when we move the particles, instead of in just a regular physiological like PBS environment, into one that has glutathione at physiological levels, 5 millimolar glutathione, there's no difference. The polymer and the nucleic acid stay bound the same way. But if the end group has this disulfide, so the whole main polymer is the same, it's just the end group changes to have this disulfide. As soon as it's in this environment of the cytosol that matches the levels of glutathione, the reducing environment, what you see is this triggered release across even a really high 200-to-1 weight ratio. It immediately releases the cargo. And so we'd have this triggered release that the particles would stay tightly bound, go through, get into the cell, get in the endosome, get out of the endosome, into the cytosol, and then know they're in the cytosol environmentally and just break open and dump all the oligonucleotide out to have this triggered release. And so that's what we wanted to do. And so we made these particles, and this is what the nanoparticles look like. And then we found that they worked really well for siRNA delivery. Here we're looking at GFP positive human glioblastoma. And so it's constitutively green. We deliver the particles with scrambled control. Everything looks the same. There's no toxicity, but the green signal stays. But then we deliver the siRNA against GFP, and it turns that signal off. And so we get this really good knockdown through these bioreducible nanoparticles. In contrast, non-bioreducible polymer at really high amounts can cause toxicity, where you see with the control that it dies too. And then in lower amounts isn't able to get good delivery, and you see all the GFP signal stays there. So with bioreducibility, it improves the efficacy, and it also further improves the viability and reduces any potential toxicity because everything degrades right away, and you can even use higher amounts of polymer. With questions, is it better to do it at the end or as we go? What would you prefer, Ali?
We usually do it at the end.
Okay, so please hold questions to the end. And I look forward to them after. And so then the question was, well, how do we want to use this technology to make a big change? And as you may know, with glioblastoma and with many cancers overall, a lot of the conventional treatments, surgery to resect them, chemotherapy, radiation, works well on the bulk of the tumor. But then there's these resistant cells that are very stem-like, brain tumor initiating cells or brain cancer stem cells. And those conventional therapies kill all the bulk cells, but then these stem cells remain to regrow the tumor. And you might not even see these by MRI. It'll look like everything is gone, but they are still there, and then they'll grow the tumor. So what we wanted to do was to design a biological therapy that focuses on just these stem cells and then causes them to differentiate and to die. And so it's something where the nanoparticles themselves could cause those stem cells to then be able to stop growing and die, apoptose, or that they also make them now more sensitive, like the regular bulk cancer cells, to conventional chemotherapy and radiation. But the critical idea is, instead of having just another therapy that kills the bulk of the tumor but leaves behind the worst parts, let's design a biological therapy that just gets to those worst parts, those cancer stem cells, and turns them off. And then would that enable then the tumor all to die, and there won't be an ability for it to regrow. And so this is what the nanoparticles look like, the microRNA with the bioreducible R646 polymer. It's fairly neutral, slightly positively charged. It doesn't cause significant toxicity to the cells, and it enables high uptake. And so that's all things we wanted to see in vitro. But then the question was, if we have these glioblastoma cells that then grow as these neurospheres that are very stem-like, how would it affect it? And so we're delivering microRNAs, miR-148A and miR-296, that then turn off stem cell markers. And we want to see, can it penetrate this neurosphere and really have an effect? And so what we find is we deliver the microRNA, we see that it is being expressed. We see that then the cells stop growing like these neurosphere oncospheres, that they have this reduced, delivering one microRNA or the combination, they have this reduced ability to grow as spheres, especially as large spheres. It pretty much turns all of that off. And then if we look at what's being expressed by those glioblastoma stem cells, we see that by delivering the nanoparticles with the microRNA, it turns off the different markers for stemness. SOX2, NANOG, BMI1, ALIG2. And so the hypothesis we had, if we deliver these microRNA with nanoparticles, it would be able to get into them and it would be able to turn off their stemness. In vitro or ex vivo, that's what we're seeing. So then we wanted to go in vivo. And this was in collaboration with John Laterra at KKI and Hopkins, where what we did is we injected locally into the tumor. And this is showing the fluorescence of how the nanoparticles spread out. They spread out through the tumor. And then if we look at the tumors with the different treatments, when we deliver microRNA that they're smaller, the tumors shrink in size. And with the combination, in most of the animals, the tumors actually fully shrank away. And so what we saw was long-term survivors. The majority of the animals, about 70%, were long-term survivors. And when we sacrificed those animals and looked at their tumors, most of them, the tumors were all gone. And so this was really dramatic. Usually with glioblastoma studies, what you see is that you see the same kind of survival curve, but it's just shifted over. You know, maybe it's shifted over a few weeks in an animal like this, or a few days. And there's some benefit, but the tumor keeps growing. But in this case, the tumor actually fully melted away. And it's because we turned off the stemness ability and the ability to regrow. And so what that meant is then the tumor cells would all just die. And we've done some other work where we combine it with radiation, and we see that this approach also makes it much more sensitive to radiation. And so this is how we can deliver these oligonucleotides and an area that we're really excited about in terms of changing the programming of cells by targeting the cancer stem cells. So those were examples with nucleic acids. Those are both negatively charged, either a large negatively charged molecule like a plasmid or a small negatively charged molecule like microRNA. But what about other things and biological drugs, biological treatments that aren't necessarily nucleic acids or negatively charged? And so we went back to our polymer approach, and this time, instead of adding bioreducibility as the key thing, what we wanted to look at was to add branching and to have more N groups, and then to look at N groups that then would be negatively charged. And the idea is that in the polymer with some positive charge and the N groups with negative charge, there'd be a balance of positive and negative charge. And that would be able to interact then with most proteins through different electrostatic interactions, through hydrogen bonding, through other interactions, that these polymers could then encapsulate a wide range of cargos of different sizes and charges. And so that was the hope. And using the same kind of approach where we make single carbon changes between the monomers that make up the polymers, we looked at the effect of this chain length to the acid group, which would be negatively charged, of 1 carbon, 3 carbon, 5, 7, 10, to see what we like the most. And this intermediate one, C5, we end up liking the most, and I'll show you. And so we wanted to make sure that these polymers could buffer the endosome, because for delivery of a protein, this is if not something like an antibody that we want to just bind to a receptor on the cell surface. This would be, well, what if we want to deliver an antibody that can get into the cytosol and then hit a target within the cell? And so we want to be able to do delivery, intracellular delivery, into the cell, out of the endosome, into the cytosol. And so we need to have the ability to get out of the endosome. And so that's where the buffering ability comes in. And so having the polymers be able to buffer is very important. And so that's what we're doing here with this titration experiment, is seeing that in the range, the physiological range between 7.4 and 5, that we're having good buffering by these different polymers. So that was important. And then we want to make sure when we add protein, they form small nanoparticles. And then that's what we see, that the protein gets complex together with the polymer into these nanoparticles. And that with C5 and C7, we see really the best in terms of the smallest size and pretty neutral charge. And so these are the kinds of attributes we're looking for in our nanoparticles. And then we looked at a range of different cell types to see if we could have this delivery. We're using here a FTSE labeled BSA. So it's albumin as a model protein. And then what we find is when we add the FTSE BSA to cells, you don't see any uptake, because it just doesn't get taken up, or anything gets taken up gets spit back out again. But if we simply just mix it with our polymer, with the new C5N group, to give it some negative charge too, what we find is that it encapsulates the protein, it delivers it into cells, and then while there's some of it that's trapped in endosomes that you see from the punctate green color, you also see a lot of it gets into the cytosol, and that's why you see this green color all through the cell. And that's showing this widespread delivery. And so what we found is that, yes, this works, and it works in a range of different cell types. And then C5 was our favorite. Well, what about different protein cargos? And so other than looking at BSA, which is large, 66 kilodaltons, we wanted to look at other ones that are even larger, like IgG, 150 kilodaltons. We wanted to look at smaller cargos like GFP or saporin. And we wanted to look at ones that had different charge, whether the charge was negative or positive or neutrally charged. Would these all be able to be equally encapsulated into the nanoparticles by simple mixing with the polymer, and then add them to cells? And so that's what we find. We find that we can deliver all of these agents. We see this good cytosolic delivery, IgG, of GFP, of saporin. And with saporin, so this is something, it's a ribosome inactivating protein. And so if it makes it into the cytosol, it'll kill the cell. But if it's not in the cytosol, if it's just in the endosome, or if it doesn't get in, or it's recycled back out, then nothing happens. And so you add the drug to cells, nothing happens. But if you mix it with the polymer added to cells, then you see all the cells die. And the red is showing dead cells too here. And so what it can do is it could be a new way to deliver cytotoxic agents to cancer cells. If you have targeted delivery to the cancer cell, this could be a very potent way to kill cancer cells through new mechanisms. Or we can look at new kinds of delivery of antibodies to different kinds of targets, intracellular targets that you couldn't really reach before. And so this opens up delivery to biologics broadly. And then an area that's interesting to think about, if we can deliver protein, can we deliver protein and nucleic acid together to do gene editing? And so we wanted to use these C5 nanoparticles with Cas9 RNPs to see if we could do gene editing. And so what we found is RNPs on their own, the cells stay green, nothing happens. But if we're then targeting that GFP and we mix the RNP with the polymer and add it to cells, then we see this really good knockout. And so depending on the cell type, it's 50% or more knockout just by, again, mixing the polymer. It could be really any protein nucleic acid. It just gets co-encapsulated together in these polymers, has very efficient intracellular delivery, efficient escape from the endosome, efficient release and degradation, and then your biologics are right there to have their effects. And then we were interested if we could do editing where we knock in as well. And we tested that out, and we could get fairly good. So total editing 40%, but HDR 4%. We could get this repair. And so it's something that this opens up another approach of having non-viral gene editing that could be done in a way that could be precise to certain cells, and then precise with how the genetic change is made to enable personalized medicine. So that was a talk about these different cargoes. And the last story that I wanted to highlight was then work that we did on using this kind of approach now. So if we can deliver nucleic acids or proteins or biological cargoes in general, what would be an interesting approach for this? And our lab's very interested in cancer. We do a lot of work in collaboration with different oncologists. And so the approach was to think about how can we do DNA delivery to enable stronger cancer therapy, and that we wanted to leverage immunotherapy. And so immunoengineering is an area that is of growing interest in my lab. As Ellie mentioned, that we now have at Hopkins a new center for translational immunoengineering that's NIH funded through a P41 that John Schneck's the director of, and I'm the associate director. And we're really interested in collaborating with others in this emerging field of immunoengineering with how we can develop together new kinds of technologies to modulate immune response. And there's a lot of different areas that are interesting, autoimmune disease, infectious disease, but one of the areas that's also very interesting is cancer. And so one of the approaches we look at in our lab is how do we make different kinds of therapeutics that are biomimetic, that mimic nature. And so with the gene therapy, we look to a virus and how can we make a small particle that has the same kind of attributes and delivers nucleic acid. In some other work, we're looking at a antigen presenting cell, and we're looking at how do we make particles that mimic features of an antigen presenting cell to have similar proteins on their surface and be able to interact with the T cell. And so in this project, what we wanted to do was to look at the APC and see how to mimic that in the body. But instead of introducing a biomaterial to mimic the cell, what we wanted to do was to reprogram a cancer cell to then act like an antigen presenting cell. And so the antigen presenting cell has a signal 1. This gives it the specificity of what antigen it's telling the T cell to go after and destroy in this case. It needs a co-stimulatory molecule that activates it and tells it to destroy. And then it also uses these signal 3 secreted molecules to help the T cell know what to do and to be properly activated. And so what we wanted to do was to have a cancer cell coordinate all these responses to train T cells to attack it and other cells in the body that have the same kind of patterns on their surface. And so we wanted to induce these tumor cells to express both the co-stimulatory molecules on its surface, as well as secretable cytokines to enable the T cell activation. And so what we're doing, instead of having the antigen presenting cell activating the T cell, we add some nanoparticles to the tumor, and then the tumor, this tumor-derived, we say tumor-derived antigen-presenting cell, the tumor is now acting just like a biologic antigen-presenting cell, like a dendritic cell, in how it can activate a T cell to then divide, and go after those cancer cells wherever they are in the body. And so this is a challenge with drug delivery and nanomedicine in general for cancer, is if you want to hit every cancer cell, that's really hard to do because you have to hit each one. But if instead you could just have a treatment that affects a portion of the cancer cells, and that could activate an immune response that then amplifies and goes through the whole body, searching and destroying cancer cells, that would be so much stronger. And so that's what we're trying to do here, is that the nanoparticle delivery to the cancer cell only has to work in a portion of them to have this profound effect. And then what about in the case where there isn't a lot of signal 1 on the surface of those cancer cells? Because we're engineering them to have the signal 2 and the signal 3, but what if there isn't a lot of the antigen? Well, you'd still be able to stimulate the signal 2 and the signal 3, which could activate natural killer cells, which could proliferate and cause cancer killing. So you could have both the T cells and the natural killer cells activated and killing the cancer with this approach. And so we initially focused on melanoma. This is a skin cancer, and looking at B16F10 cells. We've used this type of approach in my lab with a number of cell types, where we make a library of polymers with this differential structure using the same library approach from the previous slides. And then we add them to deliver DNA that's a reporter to the cell of interest. And then we can use this library approach to select materials that are very effective, and in some cases very specific for a particular type of cell, a particular type of cancer cell in this case. And this just shows with GFP expression, what this looks like, that almost all the cells are transfected. This is in vitro, but it looks really good. And then we go in vivo, where the cancer cells are on a mouse, and we locally inject it, and we see really good expression in vivo too. Okay, so the delivery part, the nanoparticles, work well for this in melanoma. Then what we want to do is then try this immunotherapy approach. So instead of delivering GFP or luminescence with luciferase, what we want to do is deliver the signal 2 and the signal 3. And so for signal 2, we have DNA encoding 41BBL. That would be then expressed on the surface of the cancer cell. And then we have DNA expressing IL-12. That will be secreted by the cancer cell. And then for all these conditions, we add anti-PD-1 is just in the background. And so what we see is that if we look at this in vitro, we see good interferon gamma secretion. And when we look at it in vivo, we see this as well. And what we know is that we can then get expression of our different factors, and we can modulate it by changing the ratio of the two plasmids. And so what do we see in terms of tumors? Well, we inject locally our nanoparticles, and then we systemically give anti-PD-1. And so in the control case, you see these tumors grow really fast. These animals only live a few weeks and then succumb to their disease, and they have to be euthanized. With delivery of the different factors, with 41BBL, there's only a very modest change. With IL-12, we see a significant change. And then with the combination, we see the best change. So delivering signal 2 and signal 3 together is key, because now that tumor cell could act like that antigen presenting cell, like a dendritic cell. And then what we found is actually a portion of these that got the combination treatment were then long-term survivors. So it wasn't just that they live longer, it's that the tumors actually shrank away fully. And so that was really exciting to see. This is what the animals look like, is that they actually had this vitiligo type white fur patch showing this cytotoxic immune response is happening. And then we took these animals, they survived for many months, and then we took them and we added tumors to their opposite side. So they don't get any new treatment, they just get tumors again. And then compared to age-matched controls that get tumors for the first time, it never have had treatment before. What we see is those survivors resisted the tumors growing, even though they didn't get any new treatment, where the controls died again really quickly within 2 to 3 weeks. And so the survivors eventually succumbed to the disease, but there was a significance in how long they survived. And so there was this memory that they had to resist the tumor. And then if we took out their splenocytes and cultured them with cancer cells, we saw this activation. And so it's something that we were changing those immune cells in the mouse to be able to resist this cancer. So melanoma is very difficult to treat, B16F10. So then we wanted to try in another model that was a little easier, is a little bit more immunostimulatory. And that's the MC38 model. This is a colorectal cancer model. And so we did the same study design of having the nanoparticles have this combination of 41BBL and IL-12 plasmids. and then with anti-PD-1 in the background. And what we saw is that this worked very potently, that the combination made just about all the tumors shrink away really quickly. And then what we saw was then long-term survival of all these animals. And then what we did later is the same thing. We took the survivors and then we re-challenged them on the opposite side with new tumors, no new treatment, just getting the tumors, and we compared them to age-matched controls. And so just like how with the first dose, the controls died, the age-matched controls get the tumor, they die, but the survivors that got no new treatment, just new tumors, have this long-term survival, and they just kept surviving. The tumors never were able to grow big. They just shrunk away. And so what we're excited about is how we can look at this approach. And this was work that was pioneered by Dr. Stephanie Zhang, assistant research professor that works together with me, that we can modulate the immune microenvironment through direct injection of gene delivery nanoparticles. And it could have a profound effect where even if just a portion of the cells are transfected, they can then lead to a strong immune response that's systemic. And that in some cases, not just clears the tumor that had the injection, but then provides resistance against any future growth of the tumor if it's injected somewhere else. And we have some ongoing work looking at metastasis. We also see that the local therapy has a systemic effect. And so I hope I've shown how polymeric nanoparticles can be effective for delivery of biologics, including DNA, RNA, and protein. And then how we have to look at the nanoparticle engineering and optimizing the polymers and the nanoparticles to achieve this. For example, small size and degradability are important, the ability for endosomal escape. And then when we think about moving from DNA to RNA, having bioreducible linkages, and we've shown this either along the backbone or in just the end group, is important to have this triggered environmental release of the oligos into the cytosol. And then for thinking of protein delivery and delivering different kinds of biological cargoes that could be positively charged in different sizes, having this balance of charge on the polymer, positive and negative, and as well as the branching, helped with that delivery. And so you can add different kinds of structural characteristics to your polymer to give it ability to have enhanced delivery of different cargoes and also have specificity to different cell types. And so there's applications for this for long-term expression, like in the eye, to express different factors to help with retinal diseases over years. There's applications for cell programming, whether we're turning cancer stem cells off or reprogramming the cancer microenvironment to then be very immunostimulatory. And then other areas as well. And so we're definitely interested in extending this technology in different ways and different kinds of collaborations. And with that, I'd like to thank all of you for your attention. And I look forward to answering questions. And I'd like to also thank all those that do this work. I feel so fortunate to have such a great lab over at Johns Hopkins. This is our lab here. And to thank all the different collaborators, Peter Compachero in his lab for a lot of the ophthalmology work, John Laterra in his lab for a lot of the work with the microRNA and the reprogramming. And then I'm happy to answer questions and look forward to it. Thank you so much.
Super. Thank you very, very much, Jordan. Really awesome talk. So what I'm going to ask people to do, if you have questions, feel free to either put them in the Q&A chat box, or feel free to raise your hand and then I can unmute you and you can ask your question. So let's start with this question. What do you think are the main challenges and limitations of using such biodegradable polymers in clinical applications?
Yeah, so the biggest challenge, has been just the delivery efficiency. And that's something that's been very difficult. And I've actually been working on this problem a really long time. I first did polymer gene delivery actually as an undergrad at Carnegie Mellon. And I'm a big fan of undergraduate research. That's what got me excited to do research. And then I continued during my time at MIT in Bob Langer's lab as a PhD and postdoc, working on gene delivery and learning library approaches with Dan Anderson and Bob Langer. And then as a PI starting out to think of what next generation libraries and how to make new types of materials. And so it's taken, 18 years to kind of get to this point, but we're still not actually as efficient as a viral particle. The viral particle is still more efficient. But what we can do is we can manufacture now these non-viral particles inexpensively, so we can just make more of them. And because they're safe and biodegradable, we can just use more of them. And so in terms of a per dose type effect, we can now achieve the viral-like levels. But it's still not as efficient. And I think there's a lot of opportunities to still make it more efficient. Where are some of those inefficiencies? The nuclear entry and uptake is a big one. So we're delivering a lot of particles and a lot of DNA to the cell, and only a small amount is actually in the nucleus. And so there's still a big challenge there to make it more efficient. In terms of clinical, because we can scale up and store this, it is something that it is translatable now. So even though that the efficiencies could still be made better, they've now gotten to the point where it's good enough. And so I think we are going to see a lot more non-viral gene therapies get approved on the market. Like, we're at that point. One of the challenges with viruses is the ability to have different kinds of cargoes, large cargoes, in there, or to repeatedly administer. But the non-viral approach sidesteps that, so that's not really a challenge. So I think we're gonna see a lot more of this, and we're gonna see the percentage of trials shift more away from virus to these non-viral approaches, including lipid-based approaches, which also have a lot of potential. I like the polymers because of the ability to tune different structural components, but a lot of the lipid approaches are also really promising. And so I think we are going to see clinically a lot of these, including in vaccine development.
Makes sense. So we have a whole bunch of questions on the basically the particles. We have a question about the mechanism of endosomal escape of the different particles you've been using, and how does the charge or the polymer structure affects it?
Yeah, that's a great question, and we actually have been probing this in some recent papers with this GAL8 assay to look at endosomal escape quantitatively, and it's actually a barrier for a lot of delivery, including a lot of lipid materials, polymer agents like PI. It really is a big barrier. So how our polymers do it is it's a combination of two mechanisms, we think. So one is we know they have this ability to buffer, because we put in these tertiary amines, and we can do acid-base titration experiments to see how they have this buffering capacity that's really focused in this endosomal pH range. So just about all their buffering happens between pH 5 and 7.4, rather than having it over the whole range. Like with PEI, it's all focused in that physiological range. And so the buffering is important, because when the protons are pumped in, it doesn't acidify and degrade the contents. Instead, the polymers become more and more charged. So they just get more positively charged. Now, to maintain electroneutrality, chloride ions, which are present throughout the cell, efflux into the endosome to balance the charge, because otherwise it would just be so positively charged. The negative ions are attracted to that. Now it's neutral. But it's a lot more salty, hypertonic in that endosome with all of these salts now in charges. And so this causes osmotic pressure. So water rushes in, and then this causes endosomes to swell. Now, when they're swollen like this, it then is something where the polymers are a mix, the ones we're using are a mix of hydrophobic and hydrophilic. And so they'll associate with the membranes of the endosome. And then as they become more and more charged, that'll destabilize the membrane. So the membrane's getting stretched, and then it's having these positive charges. So it's kind of like a overfilled water balloon, and then these needles that poke it, and it'll rupture, and that'll let the particle out. And so it's the balance of hydrophobic, hydrophilic character, and it's having the titratable groups in the physiological range of pH 5 to 7.4. Those are the key attributes that we find lead to efficient endosomal escape. And we've developed this assay. So the assay initially was pioneered by Craig Duval, and we've developed it further with different cell lines to then be able to probe this endosomal escape mechanism. And so checking out some of those papers, if you want to try that assay, or let me know, I'd be happy to share. But that's how we're probing it, and those are the features we think are important for these non-viral materials to have endosomal escape.
Gotcha. And also we have a question about the protein corona and how it affects the targeting of the cancer cells or other things that one tries to target.
Yeah, that's a really great question too. And so the examples I showed today are all about local delivery, right? So suprachoroidal delivery is in the eye, the local delivery right into the tumor, or the local delivery right into the brain. And so in those cases, the protein corona doesn't have as much of an effect because we're putting the particles where we want. And for those kinds of diseases, and that's where I see the first clinical successes with these non-viral gene therapies being is with these local administrations. You're getting really all your, big bang for your buck because you're putting all the particles right where you need them. But this is a big question for other kinds of diseases. You want to inject the particles systemically and have them find the right target cells and tissues to promote healing, to kill cancer cells. And so we're working on that. And so what we're finding is that by making these small changes to the chemicals on the end groups of the polymers, those affect the chemicals that are then displayed on the surface of the particles. And this changes the type of proteins and the amount of proteins that then associate with protein corona on the surface. And so this is some work that is ongoing in my lab, and we're varying then the polymer structure on the surface and finding that it modulates the proteins on the surface, and then we're finding that that modulates uptake into cells by different pathways. It could be like clathrin-mediated uptake versus caveoli-mediated uptake, that it'll change the route by making these small changes to the chemistry on the surface. And then in vivo, that it changes the tissues that then we see uptake and transfection happen following IV administration. So it is a really important problem. We're investigating it, and we're finding that it is, these dramatic differences. Right now, we're using it to kind of leverage, okay, well, here's a good material for this application because we have a lot of transfection of the spleen, or we have a lot of transfection of the liver or of the lung, or wherever we're seeing that. But we're trying to figure out more mechanistically to drill down exactly what are those mediators, what type of chemicals on the surface lead to which proteins, which lead to which uptake. And so I think that's a really important area of nanomedicine that a number of labs are looking at. It's a really good question.
Wonderful. And regarding the nanoparticles, what's the typical loading efficiency of them? And have you also looked at the surface chemistry and see the effects on the delivery?
Yes. So in terms of the loading, so what we do is we actually use an excess of polymer, and it's just a self-assembly process. So when we're doing, let's say, delivery of the DNA or siRNA, we're using a weight ratio of 30-to-1, 60-to-1, 100-to-1. Like we're using an excess of polymer. And so what we find is all of the nucleic acids encapsulated. You might remember I showed that gel. I could go back to it just real fast, where you can see if the nucleic acid is free. If there's any unencapsulated, it would run on the gel. So like at 20 weight here, you can see, or 30 weight, there's some of this nucleic acid that runs on the gel. But on the amounts that we use it when there's a sufficient weight of polymer that it's all encapsulated. And so there isn't any free nucleic acid. It's all fully encapsulated. The second part of the question was about surface chemistry. And so we are looking at that. We've made modifications, for example, the polymers to have PEG or other groups on the end group to change the surface. And we've also been looking at blending lipid PEG and other molecules in with the particles to modulate what's on the surface or to add a ligand. And so I think there is a lot of flexibility and modularity to do that. As we're looking now towards the systemic delivery applications, that's something that we're finding is more important for those applications too, even more so than for the local delivery. So that's one of the things we're working on in our lab now.
Gotcha. And regarding the mRNA delivery, if I understand, there's basically modifications to the mRNA that makes it stable for delivery these days. And some of the companies in that field already have lots of the IP kind of cornered on that. So how does one try to actually translate some of these types of therapeutics?
So that's a good question. So in the case of mRNA delivery, so we're not, in this case, using a new type of innovative modification to the nucleic acid. We're using the delivery vehicle as the way to do it. And so one of the things is to use less sophisticated modifications and just have the particle do the work of the delivery. And then you don't need, then you can sidestep some of the IP landscape that would otherwise overlap. But I think probably to have the ultimate delivery system, you want to have both the best engineered nucleic acid molecule and the best engineered delivery system working together. And so that would require cross-licensing. And so it might be that company focused on making the modifications of nucleic acids interested to partner with a delivery company or technology from a university to do that part together. But yeah, to those interested in commercialization, it is something to think about is, do you have, on the one hand, IP that protects your invention, but the other, do you have freedom to operate? Or do you have other blocking patents in the landscape? And then it's either that you have some cross-licensing deal, you innovate something so you don't need that, or sometimes it's just time that a lot of, earlier inventions, by the time it becomes commercial, there's only a few years left of the patent life anyway. And so it's something that if there's a new technology just being developed, it might be able to freedom to operate by the time it reaches the clinic. So those are all, I guess, approaches to that.
Wonderful. So I want to be aware of everyone's time, and particularly yours, Jordan. It's been really awesome and eye-opening to hear all the wonderful things you're doing, and definitely we generated a lot of interest. So I want to thank you very much for your time, and thank everyone else. We will continue some of our sessions. We're going to have a fireside chat this Friday as well on minimizing animal usage in research. So I think should be very exciting. So thank you very, very much, Jordan, and we'll do our own catch-up session later.
You're welcome, and thank you for the great questions and for everyone's attention. It was a pleasure to be here with you today.
Awesome. Take care. Bye, everyone.